Hey everybody, today we're going to be talking about capillary exchange. And capillary exchange is just a fancy way of saying getting stuff out of the blood and getting stuff back into the blood. So we know that the blood carries oxygen and CO2 and it carries nutrients and all the things that we're interested in getting to our cells. So how do we get it out of the blood and to our cells? That happens in a place called the capillaries. When the blood leaves the heart, it leaves the heart in arteries. In the systemic circuit, the arteries carry oxygenated blood, and the arteries are going to split several times and get into smaller and smaller vessels until eventually they're going to end up in an arteriole, which will then direct the blood into something called a capillary. And a capillary is where the vessel is thin enough that stuff from the blood can leave and stuff from the tissue spaces can get back into the capillary. It will continue along until it hooks up to a venule, which is a small vein. It will then continue on the venous side until it gets back to the heart. So capillaries connect the arterial side to the venous side of your circulatory system. And when we're talking about things leaving the blood and going into the tissue spaces, the process is called filtration. And when we're talking about things coming back into the blood from the tissue spaces, it's called reabsorption. So capillary exchange is just the processes of filtration and reabsorption. And this has to do and depends on two different pressures. The first one is the pressure from the heart. So we know that the heart beats and standard blood pressure is 120 over 80 millimeters of mercury, which means that at the heart, the contraction of the ventricles goes up to a maximum of 120 millimeters of mercury. Now, down by the capillaries, though, we have split and split and split and increased the volume of the amount of vessels so much that we only have about 35 millimeters of mercury of pressure left from that hydrostatic pressure or blood pressure. So you can understand how things are going to be pushed out of the capillary because you're basically forcing them out by the force of blood pressure. But not everything is going to leave the blood. So let's review for a moment the things that are in the blood. So the majority of what's in the blood is water. And within that water is other stuff that's dissolved in the water. So things like electrolytes, nutrients, glucose, gases. You can also have waste in there. You also have what we call the formed elements. Now we call them formed elements because not everything in there is a cell, but some of them are. So we have white blood cells, we have red blood cells, we have platelets, which are important in clotting. And then we also have proteins. So some of the proteins in your blood are clotting factors that help the platelets clot. And these are proteins. We also have globulins. So when you talk about antibodies in your blood, that is a type of globulin. And we also have something called albumin, which is a plasma protein that is produced by the liver. So we have a lot of stuff in the blood. And if we were to draw out what it looked like, you could see you have a bunch of red blood cells. Obviously, you're going to have a lot more than this. And they're actually kind of bigger. Red blood cells take up almost the whole space of a capillary, but I'm not drawing to scale here. We also have white blood cells, which are even bigger than the red blood cells. There's not that many of them. And then we have all the other stuff, the water and the nutrients and the small electrolytes and oxygen is carried in the blood. So all that stuff is in the blood. And so when you're pushing out things from the blood, really only the things that fit are going to leave the blood. So the electrolytes, the water, the nutrients, the gases, those things will fit out and be able to pass across the walls of the capillary. But certain stuff these things and these things don't fit out, so they stay in the capillary. So now you have a situation where the water is being forced out with some of the stuff, but a lot of the stuff is left in the capillary. And so there's a certain amount of inward pressure as well, and we call that colloid osmotic pressure. And it's just the pressure of a gradient. If you have a lot of stuff still left in the capillary, it's going to cause the water to want to go back in there. So on the arterial end of the capillary, that pressure is about 24 millimeters mercury. So we have a pressure out of 35 and a pressure back in of 24. So if we do the math, you have a positive pressure of 11 millimeters of mercury. So that means this one is bigger than that one by about 11 millimeters of mercury. So we're going to get a net pressure moving out. So we're going to have more filtration going on because this is higher. Now, on the other end of the capillary bed, however, we've lost a lot of our hydrostatic pressure. Remember, this was pressure due to the heart contracting. So our hydrostatic pressure on this end has fallen to about 16 millimeters of mercury. And it's just because it's that much farther away from the heart and we've lost a lot of blood volume. However, that colloid osmotic pressure hasn't changed. It's still 24 millimeters of mercury. Why is it the same? Well, because the stuff that was stuck in the capillary at this end is still stuck in the capillary at that end. So if we do quick math here, 16 minus 24, we end up with negative 8 millimeters of mercury force, which means the force moving back in, pushing water back in, is actually going to be bigger, and you're going to get a net movement back in. So you're going to have more reabsorption on the venous end than you're going to have filtration on the venous end. So what this actually means for you, I'm going to do it in a different color, and I'm probably going to make people crazy because I'm drawing on top of what I already have. What this means is that on the arterial end, water will move out of the blood into the interstitial space and will bring with it things like oxygen and nutrients, things that these cells that are living around the capillary need. And then on the venous end, we're going to have a net movement back into the capillary bed. And so anything that has built up in the interstitial space, like CO2 and other types of waste, are going to be pulled back into the capillary. They're then going to go back into the venule and head back into the systemic circulation to either be reoxygenated or excreted or filtered out in some way in some other organ. So in this way, the change in hydrostatic pressure across the capillary bed